I'm going to start this video doing something a little bit strange. I'm going to flip this coin 100 times and keep track of how many heads I get. Then I'm going to repeat this process 10,000 times, keeping track of the number of heads I get of each 100 coin tosses. Tails. Tails. Well, I'm not going to do it personally. That would require a million coin flips, and frankly, I have much better things to do with my time. But I told my computer to do it for me. For each 100 coin flips, you expect to get 50 heads and 50 tails. But sometimes you'll get more, and sometimes you'll get less. And if you look at a histogram of the number of heads of each of the 10,100 coin flip simulations, the results look almost exactly like a bell curve. Why this happens is a surprisingly deep result from mathematics and probability theory that's well beyond the scope of this video. But as it turns out, it plays an important role in our understanding of how genetics work. So let's put a pin in this whole bell curve thing for a minute, and let's talk about genetics. If you've had any exposure to genetics, you've probably heard of Gregor Mendel and his peas. I'll briefly review the key findings, and if you've already heard this 100 times, just bear with me a minute. Among other experiments, Mendel examined how the color of parent peas determined the color of their pea children. Mendel had lines of purebred yellow and green peas. Crossing yellow with green yielded more yellow peas. But when he inbred these peas together, he found 25% of the peas came out green. From this experiment, Mendel came to several profound conclusions. Most importantly, he showed that genetic information for traits passes through generation as discrete factors, genes, coming from each parent. The color of each pea is controlled by a single gene. Peas, like humans, have two copies of each gene. For Mendel's peas, the color gene came in two varieties called alleles, a yellow allele and a green allele. We call the combination of alleles an individual has its genotype. When Mendel crossed the purebred yellow and green peas, yellow peas had two yellow alleles, yellow-yellow, and the green peas had two green alleles, green-green. Individuals with two of the same allele are called homozygotes. When sexually reproducing organisms have offspring, their children receive one allele from each parent. The purebred yellow peas were yellow-yellow homozygotes, so they always passed down a yellow allele. Similarly, the purebred green peas were always green-green homozygotes, so they always passed down a green allele. So the first generation of pea offspring had one yellow and one green allele each. Individuals with two different alleles are called heterozygotes. The heterozygotes were yellow. Why yellow instead of green? Well, not all alleles are built equally. Some alleles show their effect when an individual only has one copy of it. This is the case for the yellow pea allele. We call such alleles dominant. Peas appear green only when they have two green alleles. Such alleles are called recessive. When the heterozygotes were inbred, each offspring received one allele from each parent. Which allele each parent passes down to its offspring is determined by chance. In essence, a coin flip, passing down either the yellow allele or the green allele 50% of the time. Then, if you do the math, each pea had a 25% chance of having a yellow-yellow genotype, a 50% chance of having a yellow-green genotype, and a 25% chance of having a green-green genotype. That's why about 25% of the peas came out green. And so it was undeniable that genes work in discrete units. You either have an allele or you don't. For a given gene, you might be one of the homozygotes, you might be a heterozygote, but you're in one of these discrete categories. This posed a problem for evolutionary biologists in the early 20th century. It seemed difficult, maybe impossible, to reconcile the discrete nature of genes with the observed traits of nature. For example, traits like human height had been convincingly shown to be inherited and therefore determined by genetics. Tall people tend to have tall parents. But contrary to the discrete categories implied by Mendel's peas, there aren't discrete classes of tall and short people. People exhibit a continuum of heights, ranging from short to tall. If you plot the distribution, a histogram of people's heights, it looks like a continuous bell curve. Many traits look something like this. This seems like a pretty serious inconsistency. Can we get from discrete genes to continuous variation in traits? Or is something else going on? Was Mendel wrong? In 1918, a young mathematical biologist named Ronald Fisher proposed a brilliant solution. And the answer? Well, it turns out traits like human height aren't determined by a single gene. Many genes contribute to complicated traits like height. Putting it crudely, there are many alleles associated with greater height. The more of these alleles you have, the taller you tend to be. Now, let's think about how coin flips might factor into a trait like height with a simple imagined scenario of a population of people. Using this population, let's see if we can understand why height looks like a bell curve. Imagine each person has 100 genes that determine their height. In reality, we don't know the real number. Each gene has two alleles, big H and small h. The big H allele makes you taller, so everyone has 100 genes that relate to their height, and for each gene, they have big H, small h, or small h, small h. For simplicity, let's assume the taller allele, big H, is dominant, so big H, small h, and big H, both contribute the same amount to a person's height, just like the yellow allele in Mendel's peas. Then, an individual's height is determined by the number of big H and big H small h genotypes they have. People in this population have children. Then we ask, how tall will the next generation be? More precisely, we need to ask, what does the makeup of the genotypes that control height look like in the next generation? Like their parents, each child has 100 genes that determine their height. Each child receives one allele from each parent for each gene. For simplicity, let's assume that height in no way determines who has a child of who, and that the number of big H alleles and small h alleles is equal in the population. These assumptions are not necessary to make, but they make things a little bit easier to talk about. Then, the alleles a child receives for each gene is, basically, a coin flip. Two random people have a child. Some people are homozygotes for a given height gene, some people are heterozygotes. But, on average, if you randomly pick two people to have a child, there's a 50% chance each parent contributes either a big H or a small h allele to a single gene. This is because it's assumed that there's an equal number of big H and small h alleles in the population. Then, the genotype for each child, for each gene, acts like the inbred cross for Mendel's peas. There is a 25% chance of big H, a 50% chance of big H, small h, and a 25% chance of small h. This means there's a 75% chance a child has either a big H, or big H, small h genotype, which contribute equally to height. So, what does this mean for the distribution of genotypes and height for the next generation? This situation might seem familiar to you. Each person's genotype is basically a coin flip, with a 75% chance of being big H, or big H, small h. Each person has 100 height-related genes, so it's like each person's height is determined by 100 coin flip outcomes. With that in mind, what would it look like if we plotted a histogram of the heights of 10,000 people from the population from this new generation? If you think about it, this is identical to examining the number of heads from 10,000 different 100 coin flip simulations, which, as we talked about at the start of the video, looks like a bell curve. Therefore, the histogram of heights should also look like a bell curve. And there you have it. Traits like the color of Mendel's peas and continuous traits like human height can both be readily explained by discrete genes. There is no inconsistency. In fact, in retrospect, it's exactly what we should expect. Of course, this is an oversimplification. For example, not all genes follow the simple dominant-recessive paradigm, and the frequency of each allele in the population is likely not equal for each gene. In addition, not all genes contribute to height equally, so each big H allele probably contributes differently to height. Genes can also be linked, which complicate matters. It's also assumed that height has nothing to do with who reproduces with who. In human society, that's probably not the case, and environmental factors like diet also affect height. These factors complicate the biology and the math. We'd have to do some considerably more complicated coin flips, and because of factors like these, most traits are not perfectly normally distributed. But generally, the mathematical convergence of many individual discrete events to a continuous bell curve, a normal distribution, holds strong and underlies what we observe in many traits we measure in humans and beyond. When I first learned about this, it kind of blew my mind. Here we had what seemed like a genuine inconsistency about genetics. When I first learned about Mendelian genetics, this was the first thought I had. And yet, with a little math, the inconsistency disappears entirely. It was enough to convince me that a mathematical basis is necessary to make sense of the natural world. Thanks for watching the video. I hope you enjoyed and learned something. If you did, think about subscribing to the channel. I'm excited to share more videos like this, where I talk about some of the most exciting and important ideas in evolutionary biology.